So we'll be doing renal physiology, a little bit about fluid compartments, just to see how water kind of moves between compartments and how the kidney regulates water balance in the body, and also acid-base balance, homeostasis in the body. We did a bit of acid-base homeostasis when we were doing the respiratory system. Remember, we had hyperventilation and hypoventilation, so we'll revisit that, and then we'll see how the kidney also helps, okay? So now we're going to start with renal physiology. And as always, I really like you to pay attention to the images and the PowerPoint slides. I tried to kind of find images which will be more depictive of what I'm going to say, so rather than just read. If you look at the images, it helps you. So if you look at this, you can see the three processes that take place in urine formation are all mentioned here. The first one is filtration, and where you have the glomerular filtrate being formed. Second is what is called reabsorption, and the third you see is secretion. And if you pay attention to this image, you can see how filtration, we'll see, occurs into the glomerular capsular space, and what you get is called the filtrate, which travels down the entire renal tubule or the nephron. And then we have something called reabsorption, where you can see from the tubule it's going into the bloodstream. So it's reabsorbed into the bloodstream. And when you look at secretion, look at the direction. It is in the opposite way. So it's from the blood into the tubule. And we'll see why this happens, okay? So let's see how urine is formed. When we looked at the anatomy, you remember we saw the glomerulus. We talked of something called the filtration membrane. Remember, I gave you that example of a baseball glove, and the baseball being the glomerulus, the glove being the Bowman's capsule, and the filtration membrane, which was formed between the glomerulus and the visceral layer of the Bowman's capsule. The visceral layer has special cells. Those cells are known as podocytes. So in the visceral layer, actually the cells of the visceral layer, which are squamous cells, they just have a special name. They are called podocytes because they have foot processes. So they kind of stick with one another. So imagine the cell is like this. It's a squamous cell, but it's got lots of processes like these, and these processes kind of go with one another like that. That's how they form the layer. So here is the filtration membrane through which the filtrate passes through. So blood, the filtrate is plasma without the huge proteins and huge cells which don't pass through this filtration membrane, because the pores in this membrane are rather small. And then that passes into the entire renal tubule. But all important substances are also passed through that filtration membrane, because they're just small molecules and they can make their way through. For example, you might have glucose, you might have sodium, calcium, vitamins, all of that, okay? So then it's filtered through because the pores are small and these can pass through. But you don't want it to pass out of the body. You want the body to take it back in. So then you have something known as reabsorption. And I think I mentioned this when I was talking about urinary anatomy. The reason we call it reabsorption is because absorption already occurs in the digestive tract. Remember, after digestion, everything is broken down into its smallest particles, and absorption takes place in the small intestine through the villi and microvilli. And so that absorption has already occurred. And remember how all the nutrient-rich blood went into the liver for processing, and then it passes out back into the bloodstream. Now, all of that blood is coming into the kidney. So now the kidney is kind of again taking back this. So that's why we call it reabsorption. We don't say absorption, okay? And then you have certain substances which you will see, which may fail to be reabsorbed. And so hence you need to kind of push them back into or fail to be filtered. You need to kind of push them back into the filtrate and pass them out of the body. So we have another process called secretion, and we'll be looking at each one of these. The purpose of urine formation is to regulate the volume and composition of blood. So, it maintains homeostasis. It makes sure your blood volume does not fall too low or blood volume does not rise, and then the composition of blood is kept constant, which is why it needs to reabsorb substances and remove wastes. So let's look at some of these terms. So one is filtrate, as I mentioned, is plasma, which is minus your large cells and proteins. Cells and proteins are normally not seen in urine. If you take a urine sample and you test it, the sample should be straw colored, and that is because of that bilirubin pigment which becomes urobilin. But you should see just maybe a couple of cells at the most, but otherwise you should not, and those cells come from the tubules. But otherwise, you should not see RBCs, WBCs, or anything. You should not see large proteins. If you see them, obviously there's a problem, and most of the time it would be at the filtration membrane. That means these substances have been able to pass through, okay? Urine, as you can see, contains waste. The amount is much less than what is filtered. Everything that is filtered is not passed out as urine. The amount that's passed out as urine is much less. That's how the kidneys regulate volume of blood. So if your blood volume is very low, you'll find that the urine you pass out is very little. If your blood volume is very, very high, for example, if you drink a lot of water or you're given an intravenous drip and you're given more than you need, your blood volume will go up, then you'll find that you'll pass out more urine, okay, which stands to reason. That's how it's going to maintain it. So here are the three processes we look at when we do urine formation. The first is blood is filtered. It's filtered at the level of the glomerulus, and the afferent arteriole, if you remember, the afferent arteriole had a larger diameter compared to the efferent arteriole. So as blood is passing through up here, it gets in much faster but gets out much slower, builds up a pressure here which is called the hydrostatic pressure. And through the filtration membrane, and this is the filtration membrane, which consists of the squamous endothelial cells of the capillary, the fused basement membrane, and those epithelial cells or podocytes of the visceral layer of Bowman's capsule. So through that, it comes into the Bowman's space or capsular space. This filtrate is then passing through the rest of the renal tubule, so through the proximal convoluted, loop of Henle, distal convoluted. As it's passing through, some of the important substances are reabsorbed because they got filtered, but you still need them. So they are reabsorbed, and look at the direction. It's going from the tubule cells through the interstitial space into the blood vessel. So you can see that. So it's going into the blood vessel. Some substances may not have been filtered, but your body doesn't need them. They are waste substances. So what happens is they go like this, so they don't get filtered, but your body needs to get rid of them. So it has to have like an oops mechanism. Oops, I forgot to filter it, but I need to get rid of it, okay? So how can it do that? It does that by secretion. What secretion does is, and you remember in anatomy, we saw these blood vessels were in close association with the renal tubule. So as that same waste product is passing through the blood vessels here, it actually pushes it from the blood vessel into the interstitial space, into the renal tubule. So it actually actively secretes it, pushes it out. So that process is called secretion. You can see the direction of secretion is opposite to that of reabsorption, okay? This is also one of the ways some drugs, whenever you give drugs for urinary infections, if someone has a urinary tract infection, the infection is lying here inside the renal tubule, or it could be lying in the ureter or in the bladder. But let's say it's lying in the renal tubule. You need the active antibiotic to be acting in the tubule. So drugs can be secreted into the tubule, and there they will act on the bacteria present, and that's how some of these drugs tend to color the urine a different color. Remember I said you don't call that hematuria, it's just that some drugs tend to color urine, especially drugs which we use for urinary antibiotics which we use. So let's take a look at some questions now. What is the purpose of urine formation? Very good. Number 4 is the correct answer. It does get rid of waste. it does retain important substances in the blood. Both of these together really are included in this. When you regulate the volume and composition of blood, because in the blood you don't have waste, you kind of get rid of them, you also have important substances. This answer kind of takes care of both these statements, okay? So this last statement is the correct answer. So let's look at the first phase of urine formation, which is filtration. This is dependent on pressure which is present in the glomerular capillaries. And when we did blood, if you remember, we did something called osmotic pressure, and we did something called hydrostatic pressure. Hydrostatic pressure is fluid pressure. Osmotic pressure is pressure, and fluid pressure tends to throw water out. Osmotic pressure is pressure which is, if you remember tonicity, if there's something called hypertonic, it has a higher osmotic pressure, draws fluid in, okay? So there's always a play between hydrostatic and osmotic pressure. Whichever pressure wins, the direction of water movement will be in that, along those lines, okay? So let's take a look here. So if we look here at blood passing through the afferent arteriole, thicker, larger diameter, efferent arteriole, smaller diameter. So as the blood flows into this part and it's coming out, the pressure inside the glomerulus, the fluid pressure is very, very high because it's going in easily but getting out with a little bit more difficulty. So pressure is very high. So we'll say that the hydrostatic pressure is about 55 millimeters of mercury. This has a tendency to throw fluid out. At the same time, blood has proteins inside it. Proteins and electrolytes, which are known as solutes, they make the blood, they give the blood a certain amount of tonicity. So they give the blood a certain amount of osmotic pressure, which tends to draw fluid back in. So osmotic pressure is at about, say, 30 millimeters of mercury. So it will tend to draw fluid, whatever fluid there could be present in this Bowman space, it will tend to draw the fluid back in. Between the two, which one will win? Hydrostatic pressure, right? So hydrostatic pressure will win and it will tend to throw fluid out. So because of this hydrostatic pressure, fluid is thrown out into the Bowman space. And as I said, that filtration membrane is very, very fine with tiny pores, so large molecules cannot cross it. There is a normal rate of filtration, which is about 120 to 125 ml per minute. And you can change this filtration rate. This is called the glomerular filtration rate. You can change the filtration rate. You can increase or decrease it by changing the hydrostatic pressure. So imagine, for example, during, let's say, a process where your body is in shock, or you have lost a lot of blood, or you've been vomiting profusely, or you are having severe diarrhea. You've lost blood volume. So your body kind of is stressed at that time. So the sympathetic nervous system gets activated. When the sympathetic nervous system gets activated, what is the action of sympathetic nervous system? Vasoconstriction, right? So what will it do? It will vasoconstrict the afferent arteriole. It will vasoconstrict the efferent as well. Afferent gets constricted a bit more. What happens? Less blood is going in through the afferent arteriole. Automatically, can you see your hydrostatic pressure will fall up here? Less will be filtered. If less is filtered, less urine will be passed out. So your body is not going to lose urine, and that's what you want, right? You've already lost fluid. You don't want to lose any more urine. Think of another situation. You've drunk a lot of water, or maybe a lot of alcohol, anything, lots of fluids, okay? So then you're in a nice, happy state. Your sympathetic system is not acting at that time. Here, more fluid is, and your afferent and efferent arterioles are at their regular diameter. A lot more fluid is passing through the afferent arteriole. Pressure builds up, more gets filtered, more urine is formed, and you pass out more urine. Okay? I mean, there are other things which come into play. For example, remember ADH and all that also act, which we'll see. But you can see just at this level how glomerular filtration can be affected. So let's take a look at this described.
The only blood filtration is the first of three main processes in urine formation. Blood flowing from the glomerulus exerts pressure, and this glomerular blood pressure is high enough to push water and dissolved substances out of the glomerulus and into the Bowman's capsule. This fluid then becomes known as the glomerular filtrate. The wall of the glomerulus contains pores and is similar to a strainer. The diameter of the pores determines which substances flow through and which remain. The only blood constituents that do not move into Bowman's capsule are the larger molecules such as blood cells and most plasma proteins, which exit the efferent arteriole. To summarize filtration, glomerular blood pressure causes filtration through the glomerular capsular membrane to create the glomerular filtrate.
So let's review this. So where does the filtrate first enter? So you really have to bring your anatomy and physiology together now. If you saw that video, I described it earlier as well. So where does filtrate first enter? Capsular space, yes, it does not, filtrate does not go to the efferent arteriole or the afferent arteriole. It's blood which passes from the afferent arteriole through the glomerulus into the efferent arteriole. What is filtered is called the filtrate. That filtrate first passes, as you saw in the video too, and in that other picture, it passes through that filtration membrane into the capsular space, okay? Next. Tim's kidney filtration membrane was damaged, and so here what I'm asking you is, what are the constituents of the filtration membrane? What's that? Yeah, which of the following is not involved? Yeah. Okay, few more people. Parietal layer of Bowman's capsule. And here's how, even if you forgot, here's how you should be looking at these questions, which is why I always say make sure you kind of look at images really well. Look at this part. This is the parietal layer of Bowman's capsule. So I'm going to draw it inside. This is the parietal layer of Bowman's capsule, okay? Filtration is occurring here. So it has to be the capillary, the basement membrane, and the visceral layer, which is present here around the glomerular capillaries. Parietal can't be involved in it because filtration occurs here, and that's where you find the filtrate. Look at the parietal layer. It's way outside, okay? So looking at the images often gives you an idea, so please be sure to look at these often. Okay. So here, this lady's urine showed red blood cells. What do you think this would signify? This is everything that I've already told you, so it's a review. So ideally, everybody should get this right. Very good, yes, the possibility that the filtration membrane is damaged. Okay, so you filtered this and then you're passing it down the rest of the nephron, which is the renal tubule. So now, as we said, important substances have been filtered, and you need to take them back into the blood, and that's called reabsorption. Most of the filtrate you can see is reclaimed and returned to blood, actively or passively, meaning using ATP or just a long concentration gradient, which is why you can see that your urine is less than the amount that is filtered. Urine is less than the filtrate formation. The movement is, the movement is from the tubule through the interstitial fluid, the space between the tubule and the blood vessels. That space is called the interstitial space, and it's fluid-like. So interstitial fluid, and then it goes to the blood vessels. Sometimes some substances are absorbed completely because the body needs them. One example is glucose. When you eat food and it gets digested, and the starch and everything is broken down into glucose, that is absorbed in your gastrointestinal tract, goes through, the circulation goes to the liver and everything, and then comes back through the blood to get filtered. All that glucose which is filtered, your body needs that glucose. It cannot afford to waste it. So glucose is actually, all of it is reclaimed in normal people. All of it is reclaimed, and it uses something called transporters. These are imagine little taxis sitting there which will ferry glucose from the lumen of the tubule back into the bloodstream. In people with diabetes, the reason they pass glucose in the urine, and you can understand why this would happen, is because their blood sugar levels are very high. So the amount of glucose that is filtered, is it going to be more or less? More. So the filtrate in the capsule and then in the rest of the renal tubule will contain more glucose. Now the body is going to try to reabsorb this glucose. As I said, glucose uses things called transporters, and I said they are like little taxis or ferries. There are only so many of them. So when these taxis are absolutely saturated, there are no more, I'm calling them taxis, don't write taxis near, okay? Transporters. When they are absolutely saturated, there's no more left to take the rest of the glucose. So that excess glucose, which the body doesn't need, it passes it out. And when it passes it out, remember, glucose is osmotically active. And I mentioned this when we were doing diabetes. Because it's osmotically active, it tends to draw water with it, which is why people who have diabetes tend to pass a lot of urine. And they have glucose in their urine. So that's why they have polyuria, a lot of urine, and they also have sugar in their urine. And this is the reason why it happens. You will see as we go along, the area where reabsorption occurs, the proximal convoluted tubule, is the main part where most of the reabsorption occurs. And if you remember, the cells were very tall. They had microvilli in them, a lot of mitochondria. So that would provide surface area plus energy. So a lot of other substances are reabsorbed, and that amount that is reabsorbed is called mandatory reabsorption. That means the body has to take back so much of sodium, it has to take back so much of chloride, it has to take back so much of calcium, and so on. The rest, as it is passing through the distal convoluted tubule and the late part of the ascending limb of loop of Henle, that is the area where it is dependent on hormones. Remember, we did aldosterone, which does sodium reabsorption and potassium excretion. We did parathormone, which does calcium reabsorption to raise blood calcium levels, right? Water, for example, ADH is responsible. So all of these at the distal parts of at the distal convoluted tubule and the distal part of the loop of Henle, that's where the rest of the reabsorption of some of these substances takes place, and it's dependent on the need of the body, and that is where the hormones act. Have you followed? Okay. So let's take a look at reabsorption. So let's just go and look up here. First is the proximal convoluted tubule is the main site, as I told you. When we look at this loop of Henle, there you can see there are two limbs of this loop of Henle, and they act a bit different, both of them. And it's important for you to remember the difference between these two. The descending limb of the loop of Henle, as you can see here, water is passing out. That means it's being reabsorbed. So the descending limb of the loop of Henle is permeable to water. The ascending limb of the loop of Henle, so this is descending, permeable to water. The ascending limb of the loop of Henle, notice sodium, chloride, potassium are going out. That means solutes are going out. So the ascending limb is not permeable to water. So it's only permeable to solute. So this part is pretty much impermeable to water. I got my spellings all mixed up there. So this is impermeable to water. This one. And this one is permeable to water. And we'll see later how, because of the fact that water moves out here but only solutes move out here, that the concentration in the medulla, the gradient kind of keeps changing. Then in the distal convoluted tubule and the collecting ducts and even the latter part of the ascending limb of loop of Henle, this is, as I mentioned, where hormones will act. So ADH, as you can see, if your fluid volume is low, that's when you want ADH to be secreted so that more water is reabsorbed. Aldosterone will be secreted when sodium levels are low. Parathormone is secreted when calcium levels are low so that it can reabsorb calcium. If instead your water levels in your body are very, very high, then ADH will be suppressed, aldosterone will be suppressed, and in their antagonist ANP will be secreted. So you can see that in this area is where all of these hormones will be acting. So let's take a look at reabsorption.
Reabsorption is the movement of substances out of the renal tubules into the peritubular capillaries located around the tubules. Water, glucose, sodium, chloride, and other nutrients are examples of substances that are reabsorbed during this process. There is a repeated pattern for reabsorption. First, positively charged ions, like sodium, are generally actively transported out of the tubule and into the blood. Then, negatively charged ions, like chloride, follow the large positive charge into the blood due to electrostatic attraction. Then, water follows by osmosis. In the proximal convoluted tubule, the sodium and chloride are transported out of the tubule into the blood. Then, water follows. In the loop of Henle, the pattern repeats itself. Sodium and chloride are transported out of the tubule into the blood. Then, water follows. Finally, in the distal convoluted tubule, it's the same thing again, but this time it is regulated by a hormone called ADH. Sodium and chloride are once again transported out of the tubule into the blood. Then water follows. In total, approximately 99% of water that leaves the blood by glomerular filtration returns to the blood by reabsorption. This leaves about 1% of the filtrate to be excreted as urine. Absorption occurs through active or passive transport. Sodium ions and other ions are only partially reabsorbed from renal tubules. As an example, sodium ions are actively transported back into the blood from the tubules. The amount of sodium reabsorbed varies depending on salt intake. The greater the salt intake, the less the amount of sodium reabsorption. The less the salt intake, the greater the amount of sodium reabsorption, and the less sodium secreted in the urine.
So that makes sense. The more sodium you take, there's too much sodium in the filtrate. You don't need to reabsorb so much. You have to pass it out, and aldosterone won't be secreted. Okay, so let's take a look at some questions to do with this. What is the purpose of reabsorption? Yes, the most important purpose is to reclaim important substances that get filtered. Nutrients, provision of nutrients is through digestion. That's already done its job. You're kind of redoing some of that. You're like reabsorbing to substances which are getting filtered normally. So let's look at this question. Very good. Yes, substances pass from the renal tubule into the peritubular capillaries. That's why I show you the video so that you can actually see it passing through, okay? Not that substances pass the other way around. That we'll do now, which is another process which is known as secretion. So the third process, third part of urine formation is what is called secretion. And as I said, I like to call it the oops mechanism, or it's a backup mechanism. I think your book gives a very nice description of a garbage conveyor belt. So imagine a garbage conveyor, trash conveyor belt where all the trash is thrown, okay? And so that's filtration which is occurring. And then you have all these people who are picking up some of the important things that were trash but maybe useful. Like, you know, people throw all kinds of things, but they're useful things which they throw. So you pick that up from the trash. And that is what is called, think of that as reabsorption. And then, after that, two things happen. Somebody picked up something and says, oops, oh, I don't really need it. And you throw it back into the conveyor belt, okay? So you're pushing it back, so that's secretion. Or it could be some things actually went down the conveyor belt, so you didn't pick them up. So now when you they come into a pool, you realize, oh, this is really not what is needed, so I need to kind of throw it back into the belt. And that, so you can see like it's something that didn't get filtered, and then you picked it up and you throw it back into the conveyor belt. So that is secretion. So it's kind of like, that's why I say it's like an oops mechanism. Oops, I picked this up and I really don't need it. Or, oh, it didn't get filtered, but I see that it's present here and I need to get rid of it, and so you throw it back, okay? So the process is the other way around to reabsorption. So it'll go from capillaries into the interstitial fluid and into the tubules. And the purpose is to get rid of all your unwanted substances and waste that either were not filtered or were reabsorbed just because of a concentration gradient. Your blood pH is also maintained. Remember I said that the kidneys take part in maintaining acid-base homeostasis. So you will find that some substances like hydrogen ions or bicarbonate ions, depending on whether there is acidosis or alkalosis, the blood pH can be maintained. If there is acidosis, that means there's too many hydrogen ions. So what does the body want to do? Get rid of the excess hydrogen ions, right? So if you got rid of them in the urine, and you'll see how we get rid of them, you secrete them back into the tubule so that they pass out in the urine. Or if you have alkalosis, or if you have acidosis, what you can do is reabsorb a lot of the bicarbonate and push hydrogen ions out. Or if you have alkalosis where you have too few hydrogen ions, what you'll do is you will reabsorb the hydrogen but push bicarbonate out, so that way you'll be able to maintain homeostasis. So that's how the kidney kind of takes part in this. Secretion also occurs mainly in the proximal convoluted tubule. Again, that's why the proximal convoluted tubule, the histology is relevant to what processes occur there. And the hormonal influence occurs here as well, because aldosterone, remember, it reabsorbs sodium, but it actively secretes potassium. So if there is too much potassium in the body, aldosterone will be released and it will push the potassium out, okay? So let's take a look at secretion.
Although most of the water and substances enter the tubules because of filtration across the glomerulus, the third process in urine formation, known as tubular secretion, moves a small amount of substances from the blood into the tubules. Substances such as potassium, hydrogen, uric acid, ammonium, and drugs such as penicillin and histamine move from the peritubular capillaries into the tubules.
So notice how she mentioned drugs. You know, penicillin is one of the drugs which is often given in urinary tract infections. There are other drugs which are given whenever you take a drug that's absorbed into your bloodstream. It may not be filtered, so it's kind of going back into the bloodstream. Now you can actively secrete it into the tubule so that there it can act on whatever it needs to act on. So here, there is how do you regulate urine volume and composition. So we did part of it. Also, you can see that what happens is that when you pass through, remember the descending limb of loop of Henle was permeable to water. So water kind of moves out of the descending limb. So by the time the filtrate comes down into the bend of the loop, you can see it's going to get more and more concentrated. And then when it goes up into the ascending limb, remember the ascending limb was impermeable to water. Solutes kind of moved out, right? So here now the urine starts getting more and more dilute. All of this water or solutes pass into this interstitial space of the medulla up here. So that kind of creates a concentration gradient from the top to the bottom. So the loop of Henle has what is called a countercurrent mechanism. So the filtrate gets more concentrated as you come down, and then it in the opposite, when it moves up in the opposite direction, it starts getting more and more dilute as you go up. So this kind of a mechanism then finally decides whether whether more water is going to be reabsorbed in the collecting duct, or you want to pass more water out where it will be under the control of hormones. So let's take a look at this, because this will kind of explain it to you even better.
One of the primary functions of the kidney is better understood at the level of the nephron. In forming urine, the nephron functions to remove metabolic wastes from blood while regulating the volume and composition of body fluids. Urine formation begins in the renal corpuscle, where blood passing through the glomerulus is filtered. Filtrates include water, ions, and other solutes, which are filtered out of the blood and into the capsular space to begin urine formation. Urine passes into the proximal convoluted tubule, then into the nephron loop. The nephron concentrates urine by the process of the countercurrent multiplication system. The countercurrent multiplication system is basically the means by which water and sodium are extracted from the filtrate as it passes through the tubules toward the collecting ducts. This system is called countercurrent because of the dynamics set up by the hairpin turn of the loop of Henle. The direction of flow in the descending limb of the loop is opposite from the direction of flow in the ascending limb. By flowing in opposite directions in such close proximity, a unique interaction occurs. The loop of Henle dips from the cortex of the kidney into the medulla and is surrounded by the medullary interstitial fluid. The countercurrent multiplication system is responsible for not only elevating the concentration of urine in the loop, but more importantly, it elevates the concentration of the interstitial fluid as well. To explain the countercurrent multiplication system, we must first imagine the loop filled with a stationary column of glomerular fluid, which includes water, salt, and urea. At first, the concentration of the glomerular fluid and the surrounding interstitial fluid will be equal at an osmolarity of 300 milliosmoles. Within the walls of the thick segment of the ascending limb, special pumps extract salt, sodium chloride, from the fluid and transport it to the interstitium. The two areas no longer have an equal osmotic pressure, and a gradient has been established. Within the thick segment, the concentration is at 200 milliosmoles, whereas in the medullary tissue, the concentration is at 400 milliosmoles. Over time, a net diffusion of salt into the descending limb and a net diffusion of water out of the descending limb results in a balance in osmolarity between the descending limb and the surrounding interstitium. Because the thick segment is still actively pumping out salt, the osmolarity of the ascending limb is still 200 milliosmoles, and the interstitium and descending limb are balanced at 400. Salt accumulates in the medulla while water is drawn out of the fluid in the descending limb. Water is removed by the vasorecta, which are capillaries that surround the loops. In real life, operation of this gradient occurs while the fluid is flowing. Fluid flowing in from the proximal tubule and out to the distal tubule, along with the constant pumping of salt into the medulla, contributes to the multiplication of this gradient. As fluid follows the path of the loop, an increasing amount of water is extracted as it reaches the bottom, and an increasing amount of salt is pumped out as the fluid reaches the top. The more salt pumped out, the higher the concentration in the medulla. The higher the concentration in the medulla, the more water is extracted. Eventually, the levels of osmotic pressure are equal horizontally, with the highest value being at the tip of the loop. This level is at 1,400 milliosmoles per liter, which is also the value of the maximal osmolarity of excreted urine. As you can see, fluid becomes more concentrated as it rounds the loop, but it immediately becomes rediluted as it reaches the top. Final concentration of the fluid occurs in the collecting ducts, as it must travel back through the concentrated interstitium of the medulla on its way to the calyces. Urine is concentrated even further in the collecting ducts. The collecting duct must transport urine back through the concentration gradient formed by the nephron loop. The walls of the collecting duct are permeable to water, but not to salt. Therefore, an increasing amount of water is drawn out by osmosis as the fluid travels through the gradient. The gradient provides the force for the concentration of urine, but the rate is determined by the amount of antidiuretic hormone, ADH, in the blood. An increase in ADH in the blood causes the collecting ducts to become more permeable to water. Less urine is secreted and it is more concentrated. A decrease in ADH results in a decrease in water removed, thus a larger volume of more dilute urine is excreted.
I don't want you to try and remember the numbers up there, but as long as you understood this thing, that the filtrate starts getting more concentrated as it comes down, gets more dilute as it goes up, but sodium ions keep on getting pumped here. So as water is removed from the ascending limb, sodium, because of a gradient, tends to go in. And then as we come down the collecting duct, depending on how much water there is, ADH will be secreted or will not be secreted. So here, when we look at how urine, whether you pass dilute urine or you will pass concentrated urine, it's really common sense. So think about this. When would you pass dilute urine? When your body has large amounts of water, right? Then you want to pass out that excess water. So if you drink large amounts of water, or for some reason you got an intravenous drip and you got more than normal, anything that makes you drink a lot of fluids or gets a lot of fluid inside you, ADH would automatically be inhibited at that time because you want to pass out more urine. So because the osmoregulators in the hypothalamus sense this hydration, so water has to leave the body. So the collecting ducts you saw in that video become impermeable to water. So if water cannot pass out from the collecting ducts into the interstitium and back into the bloodstream, all of that water will pass out in the urine. Got that? So ADH is what makes the collecting ducts permeable or impermeable to water. If ADH is present, the collecting ducts are permeable to water. Being permeable to water means if this is the collecting duct, these are the cells of the collecting duct, and there's water here. Being permeable, the water will pass into the cell, out of the cell into a blood vessel which is lying here. And this requires the presence of ADH. If there is no ADH, these cells become impermeable to water. They do not allow water to flow, so all this water will pass out. Okay? Let's look at the opposite situation. When do you think you'll pass concentrated urine? When you haven't drunk a lot of water or you've lost fluids through vomiting, hemorrhage, diarrhea, anything like that. So anything which causes dehydration, your osmoregulators sense that the body is dehydrated. They immediately secrete ADH. The collecting ducts become permeable to water, so you want to retain that water. So they pass from the lumen of the tubule into the cells, into the interstitium, into the blood vessel. So less is passed out, so you end up passing concentrated low volume urine. Okay? So even if you work backwards, you can see what would be secreted, when it would be secreted. And it's really common sense if you think about it. Okay? So this was to do with water. Solute, sodium and potassium sodium reabsorption and potassium secretion. Secretion of potassium is regulated by aldosterone, which is through the renin-angiotensin mechanism. If there is low blood pressure or your blood volume is low or there is less sodium or more potassium, automatically aldosterone is released, which will reabsorb sodium. Whenever, remember, sodium is reabsorbed, you saw in the video, water goes with it, so it raises your blood volume and automatically you'll pass less urine. Also through that renin-angiotensin mechanism, angiotensin causes vasoconstriction, which will automatically kind of decrease the filtration because it will cause vasoconstriction of the afferent arteriole. So again, it will decrease filtration and increase blood pressure. If, on the other hand, you have too much fluid, then ANP will, or too much sodium, atrial natriuretic peptide will be secreted, so that will push the sodium out and along with that water. pH, we will see, is regulated depending on whether there's acidosis or alkalosis. If blood pH is acidic, that means too many hydrogen ions will be secreted into the lumen and passed out. We'll see how it's passed out in the urine. If it is alkaline, you want to bring down the pH. So hydrogen ions will be reabsorbed, instead bicarbonate will be secreted. Okay? So let's take a look at this. What do you think drinking alcohol such as 3 cans of beer will do? Dilute urine to be formed. And if you remember, when we were doing endocrine system, I also told you that alcohol, not only because of volume of fluid, automatically your volume goes up and so you'll pass that out, so your urine will be dilute. Alcohol by itself also suppresses ADH formation or ADH secretion. In other words, you don't have to drink like 3 cans of beer or many large glasses of wine. Even if you take shots, just alcohol by itself, you will find that you tend to pass more urine because alcohol suppresses ADH formation, which means that more urine is formed and automatically you tend to pass more urine. Okay? So, what about this one? Roger sweating excessively. What do you think will happen? Very good. Concentrated urine, already losing water through one side of the body, one aspect. You want to conserve it from the other. Next, let's look at, so that's renal physiology, and now we're kind of going to tie that up a little bit in with fluid compartments. So when you look at your body weight, water forms 60% of your body weight. So of this 60%, you will find that the water is present either within the cell, where it is called intracellular fluid, or it's present outside the cell, where it's called extracellular, outside the cell, okay? So anything outside the cell is extracellular. Notice that the extracellular area is divided into 2 parts. One part is called interstitial, that means in between. Interstitial, and the other part is what is present inside the blood vessels. Remember, blood is a fluid, so in blood vessels, the plasma that is present, that is also part of extracellular, right? That would be part, the plasma would be part of extracellular fluid. We just put it in a separate compartment of extracellular fluid, and the interstitial fluid, which is what you saw where you had the renal tubule. So here you have the renal tubule. Here you had a blood vessel, right? And this was the interstitial space. So this is also extracellular fluid. The fluid in this is also extracellular fluid. Do you understand that? But the cells inside the blood, that means the red cells, the white cells, and the cells which form the lining of the renal tubule, all these will be part of the intracellular fluid compartment. So if you just think of it, anything inside the cell, the cytoplasm of the cell is intracellular. Anything which is outside is extracellular. So everywhere in the body you will find that cells need to be bathed by this extracellular fluid, because that's where nutrients get transported to it. So therefore, inside the blood vessel also, the cells are intracellular, and they are bathed by this extracellular fluid. Okay? So don't make a mistake between the two compartments. Intracellular is just everything which is inside the cell. Extracellular is broken up into two parts. That part which is in blood vessels, which is the plasma, and the other part which is outside the blood vessels but still not in the cell, that's interstitial. Okay? So let me go back. Okay. So when you look at composition of body fluids, whether it's present inside the cell or outside the cell, water is the main component. You have electrolytes, so sodium, potassium, chloride. Remember when you did ANP1, you did muscle physiology. If you remember a muscle fiber, we saw that there was more, normally at resting membrane potential, there was more sodium on the outside, more potassium on the inside. You remember that? Okay, so that's what we are really talking about. You have acids which are present, which we look at. We have bases. Then you also have non-electrolytes, like you have lipids, amino acids, glucose, vitamins, urea. All of these are present within and outside the cell as well. In the extracellular fluid, there is more sodium and more chloride, which should explain why when we look at a muscle fiber, this is the inside of the muscle fiber. So intracellular, we draw potassium on the inside, and we put sodium on the outside. Remember, when the muscle gets depolarized, sodium starts moving in and potassium moves out, and then you have the sodium-potassium pumps which kind of bring back everything to normal, okay? Inside the cell, the intracellular fluid, potassium is more, hydrogen phosphate is more, and you have certain proteins which are more, okay? So here is a table which is kind of showing you the electrolyte composition of intracellular and extracellular fluid. Obviously, I cannot expect you to remember all of these. So there are certain things which, whatever I've given in that previous slide, sodium, for example, is more on the outside, potassium more inside the cell, chloride more on the outside, hydrogen phosphate more on the inside. So this table will kind of explain whatever is given on the previous slide. So I'm not going to kind of pay too much attention, just compare the two. So in order to look at how fluid passes between intracellular and extracellular compartments, we have to understand hydrostatic and osmotic pressure. We've been talking about this right from blood. I also mentioned this when we were doing filtration. Remember, I said hydrostatic pressure tends to throw fluid out. Osmotic pressure tends to draw fluid in, and because hydrostatic wins, that's why fluid pressure increases. So let's again revisit this. So this was a video I'd made earlier, so instead of drawing it all over, I'm going to kind of just play the video.
Okay, so I'm going to explain hydrostatic pressure and osmotic pressure and the relationship between them. So here are two beakers. They have water, and I'm assuming they have the same volume in both beakers. In this one, it is made up of all water, and here we have partly water and part of it is solutes, for example, sodium, potassium, chloride. Those are the solutes which are present. Now here, the pressure in this beaker, which is fluid pressure, in this case I said water, is what is known as hydrostatic pressure. In this beaker, because of the presence of solutes, we have something known as osmotic pressure, and when your solutes are more, so the greater the solutes, the greater the osmotic pressure. So what it tends to do is the solutes tend to draw water towards it. So now let's see how this can change. So here I have the same beakers, and I'm adding the same quantity of water to both beakers, and we'll see what the change will be in hydrostatic and osmotic pressures. So the plus stands for I'm adding water to it. So if I add water to it, the hydrostatic pressure will increase because it's based on fluid pressure. So more fluid, more pressure. Here, when I add water to this beaker, what will happen is the solutes, the concentration of the solutes will go down. So therefore, the osmotic pressure will decrease. So when you have more fluids added, then the solutes get diluted, so your osmotic pressure decreases. So you can see that when you add water, the opposite effect happens in hydrostatic and osmotic pressure. Let's look at the other situation where we remove fluid from the beaker. So the minus stands or the negative stands for removal. So when I remove fluids, just fluid will leave the solutes behind, then it gets more concentrated. So now my osmotic pressure will go up. And if I remove fluid from the beaker, we just had water, since my fluid amount is less, so my hydrostatic pressure will decrease. So again, you can see the inverse relationship between them. And here's an example when, let's say, somebody has vomited or is having diarrhea and they lose a lot of fluids. They do lose solutes also, but let's say they lose a lot more fluid than solute. Their hydrostatic pressure goes down and the osmotic pressure goes up. And let's say you were to give somebody an IV drip and you give them more than normal, what will happen? Their hydrostatic pressure will go up and the osmotic pressure will go down. So this is the relationship between the two.
So from this, what we could say is that the more the fluid, the greater the hydrostatic pressure, right? The lower the osmotic pressure, isn't it? Because solutes will get diluted. The less the fluid, our hydrostatic pressure will drop, but our osmotic pressure will go up because the solutes tend to get concentrated. Okay, can you follow this? So let's go to look at, this is the same thing that happens here. And this was something that we did in blood, so we are kind of revisiting this. So let's look at what happens as how substances move between cells and the bloodstream. Because remember, everything is finally taken into the blood, and the blood is what takes these substances to the cells, and then it also brings back substances from the cells. So let's look at a blood vessel. So, you know, your aorta and all its branches, and then remember, they finally break up into capillaries. Capillaries have an arterial end and a venous end. The arterial end always has higher pressure. The venous end is at a lower pressure. The venous end of the capillary will join and then form small veins, which will join and form larger veins and will go up, okay? So let's look up here. So these are the tissue cells on either side. So what compartment will this be, the tissue cells? Intracellular compartment or extracellular compartment? Intracellular, right? So I will say intracellular space or intracellular fluid. Okay? This area, which is outside the cell, which compartment is that? Extracellular. So there is this part which is all blue, which is called interstitial fluid. So this is one part of the extracellular. The second part of the extracellular is the plasma which is flowing in this blood vessel. Got that? So that's why the interstitial fluid and the plasma both belong to extracellular fluid. Have you followed? Okay. So now let's look at this. This is the arterial end of the capillary. This is the venous end of the capillary. And here at the arterial end, the blood flows at a higher pressure than it does at the venous end. So let's say the flow of blood at the arterial end, the pressure is about 35 millimeters of mercury. That 35 millimeters of mercury will tend to throw fluid out from the blood vessels. Blood vessels have tiny pores or what are known as fenestrations. It will tend to throw a little bit of the plasma out into the interstitial fluid. From the interstitial fluid, that can go into the cells, okay? But inside blood, you also have electrolytes, right? Remember, you have plasma proteins, you have sodium, you have chloride, you have all those other things. So they exhibit or they express an osmotic pressure because osmotic pressure tends to draw fluid in. So let's say the osmotic pressure of blood is about 25 millimeters of mercury. So you can see hydrostatic pressure is tending to throw fluid out. osmotic pressure will tend to draw fluid from the interstitial space into the blood vessel. Between the two of them, the hydrostatic wins. So the net effect is the net, what is called filtration pressure, is 10 millimeters in favor of hydrostatic. So fluid moves out at the arterial end of the capillary. That fluid moves out and it's going into the interstitial space. It bathes these cells, and based on what the solute concentration is here, it will either pass into the cell or remain there. Let's come into now the flow kind of becomes slow and it comes into the venous side of the capillary. Since flow has slowed down, the pressure, hydrostatic pressure, is much lower. So from 35, it kind of dropped to 17 millimeters of mercury. The same amount of solutes which were present here are still going to be at the venous end. Nothing was lost. So your osmotic pressure is going to remain at 25, right? So which one wins in this situation? Osmotic pressure. So now fluid will go back from the interstitial space into the blood vessel. So you can see this is one way that fluid volume is kind of kept constant, that the arterial end fluid goes out, at the venous end some fluid comes back in. So that's how part of your blood volume is kept constant, okay? Then based on what is the hydrostatic and osmotic pressure, the amount of solutes which are present in this interstitial fluid, the water will either move into the cell or water will come out of the cell. So if here solutes get concentrated, what will happen? They will tend to draw fluid out. If on the other hand solutes get diluted here, there are more solutes, they will tend to push water into the cells. Okay, have you followed that? So having looked at that, let's kind of look at how fluid moves through these various compartments. So if you read through this, so between the plasma in the blood vessels and the interstitial fluid, remember both of them belong to the extracellular fluid space, right? So this will be across the capillary walls, and this is dependent on hydrostatic and osmotic pressure interaction, right? Because this is what we did here. So that means between the fluid, the plasma here and the interstitial fluid here, the movement will be across the capillary walls, and it will depend on hydrostatic pressure and osmotic pressure. Whichever one is greater, the fluid will move according to that. And we know that hydrostatic pressure tends to push fluid out, osmotic pressure tends to bring fluid in. The next is then between the interstitial fluid and the intracellular fluid. That means the cytoplasm within the cells will be across the plasma membrane of the cells. Correct? So again, if we go back to this, so between this, so after fluid has moved out here, then between the interstitial fluid and the intracellular, that means the cytoplasm, it will be across the plasma membrane. Correct? Water moves very, very freely across these based on the gradient that is present. But for ions and large molecules, ATP is used. So that is an active transport. And this is obvious. There's always a one-way traffic of wastes and nutrients. Finally, all nutrients have to go into the cells. Wastes have to come out of the cells and then be pushed out of the body, right? Either through respiration or through the kidneys. So therefore, if you look at it, nutrients will go into the cells. So that is nutrients, and wastes will come out of the cells and pass this way and then will be finally taken out. The extracellular solute concentration is extremely important. This determines the volume of intracellular fluid. So in other words, what I'm trying to say up here is whatever the extracellular solute concentration first is within the blood vessels, that will determine how much fluid will go out into the interstitial fluid or how much fluid will come into the interstitial fluid. And then whatever the extracellular fluid solute concentration is here in the interstitial space, that will determine what the volume of the intracellular fluid compartment is. Okay, with all this intra, extra and all that might get a little confusing, but think of it this way. Suppose the amount of solutes was a lot in this area. What would that make it? That would make this area hypertonic, right? The osmotic pressure would be more. So what would it tend to do? It would tend to pull fluid out from here, from the cells, because when you say it is hypertonic, that means it's hypertonic compared to the cells. So what will happen to the cells? The cells will shrink. On the other hand, let's say the solutes are diluted, so that means the amount of solutes is less, but water is more, so it's hypotonic. There's more water here than inside the cell, so it will push water into the cell, so the cells will swell. Have you followed that? So that's what we mean by the solute concentration here is what determines what the volume of the intracellular fluid compartment will be. So to explain this, that's not what I intended to do. So let's take a look at this with a little simple experiment for those who haven't understood this concept. This talks of osmotic pressure. So we'll see that it's the number of particles in anything which increases the osmotic pressure. It's not the size that matters. So if you have, for example, 3 really tiny sodium particles and 1 huge albumin particle, the area where the 3 tiny sodium particles, the number of particles, that will have a greater osmotic pressure, okay? So it's the amount of solutes, not the size of the solute. So let's take a look at this. So here we are beginning with 2 mesh bags, and it says imagine this to be a blood vessel, each one. And here are measuring cups holding 4 sponges each. Each color sponge represents a solute. So albumin is red, sodium is green, and glucose is blue. And we'll drag the 6 measuring cups on the left to bag A and 3 measuring cups to bag B. So let's drag these here. Now each is filled with particles which are very similar to what happens in someone's blood. So let's look at the pulling pressure now. And we drag it to the bucket of water. And now we measure how much water each pulled in. So this is 90 ml, and you can see this was 45 ml, and you can see on this side it was double the number of solutes that it was compared to this side. So A had twice the pulling power as B did. So the higher the osmolarity, the greater the pulling power, which is why we say that the amount of solutes determines what the volume would be, okay? So let's answer some questions like this. The greater the amount of particles, the stronger the pull of fluids in their direction. So if a client has a serum albumin of 1.9 grams per deciliter, would fluid be more likely to enter and stay in the vessel or leave it? So take a guess, since you all don't know the normal amount. So take a guess and we'll see what the answer is. So that means a person has so much serum albumin inside their blood vessel. So do you think fluid is likely to enter or fluid is likely to leave? Enter. So stay in the vessel. The albumin amount is below normal. The amount of albumin is much more. Even I don't remember the exact amount. So this is really low. So this person obviously probably has some sort of a kidney disease, which is why you see people with kidney disease looking edematous. They usually have bags under their eyes. Their feet are swollen because they lose albumin. So what happens is that most of the fluid tends to remain outside, doesn't go back into the blood vessels. Okay. Suppose a client had a serum glucose level of 566. Would fluid be likely to enter and stay in the vessel or leave it? So it looks like you all know the correct. Remember, anything more than 200 is considered abnormal, right? Okay. So you've understood now how the solutes act. So let's look at this. So this is what I explained. So imagine if someone is dehydrated. So there's loss of fluid from the extracellular fluid compartment. So the solute concentration goes up. So therefore the osmotic pressure rises. And this will draw fluid out from the cells and they will shrink. So look at this. Someone is dehydrated, they are vomiting. So then fluid is going from the lumen of the gastrointestinal tract going out. So that's not within the cells, that's part of the extracellular space, so it's going out. Someone is having diarrhea, same thing, right? So the blood volume comes down because of that, so automatically fluid is coming out of the extracellular compartment, solutes go up, so it becomes hypertonic, so it pulls fluid from the cells, and cells, that's why tend to shrink, which is why a dehydrated person, you notice how their skin tends to look, the eyes are sunken, the skin kind of loses what is known as turgor. Normally when your skin is healthy and well hydrated, if you kind of pull your skin up and it'll drop back to its original position very, very fast. In a dehydrated person, if you pull your skin up like this on the hand, it'll kind of stay up for some time. It loses that elasticity. Okay. Look at this other condition where someone is overhydrated. Let's say you drink water too quickly, too much water, you're drinking really, really fast and you're not passing fast enough out in the urine. Or let's say someone's got an intravenous drip, and this happens quite often in children. In the pediatric thing, you have to monitor them very, very carefully. You always give them fluid according to their body weight, because when you put an intravenous drip, and let's say the nurse is busy and she forgets, you give them more fluid than normal, you come back and you find the child a bit swollen, edematous. So you see a lot more fluid is present here, so the solutes get diluted, which means osmotic pressure falls. So now it's less than the intracellular compartment, so fluid will go into the intracellular compartment, the cells kind of swell, okay, which leads to what looks like edema. So let's take a look at a few questions here. So the first one, which of the following conditions will not increase osmotic pressure? Very good. Drinking a lot of water. When you drink a lot of water, more fluid goes in. It dilutes the solutes. So the solute concentration is low, which means that automatically osmotic pressure is less. When you vomit, you pass out a lot more fluid. Solutes also go out, but fluid is more compared to the solutes. Same for diarrhea and same for hemorrhage. The volume of the fluid, water going out, is much more than the solutes which go out. Okay? Then when we look at water intake and output and regulation of water and electrolytes, this is kind of a review because you've done a lot of it before. When you take in water, 60% of it is through ingestion of fluids. And this is common sense, if you sweat a lot, if you're dehydrated, then this loss will be through the extracellular fluid and the cells will shrink. So you can see how your average intake is 60% of it comes from whatever you're drinking, 30% comes from the food that you eat, the water inside the food, and metabolism is 10%, because remember, when glucose is broken down, for example, it breaks down into ATP, carbon dioxide, and water. So water comes from within metabolism inside the cells as well. Well, your output, if you look at it, 60% is through urine, and then you have the other remaining 700 ml and a little bit, 200 and 100 is feces, is a little 4% goes out through that, through sweating, about 8% goes out, and through losses through the skin and lungs when you breathe out the water vapors, about 28%. Some people say that they don't sweat at all. Everybody sweats unless you really have a problem with your sweat, which is anhydrosis. If you're really, you may not have very visible sweating, but some people sweat less than the others, but everybody does sweat, so you may not notice as much, but you do have that insensible loss. Now, this is what is called, this loss which occurs through the kidneys, lungs, and feces is what is called obligatory loss. We have to lose this amount of fluid. The rest of the water loss depends on how your body's needs are. If you drink more, if you have taken more fluids, automatically your water loss will be more. So your urine becomes more dilute. Remember, you pass large quantities of dilute urine. If you drink less because of, let's say, you're sweating a lot or your intake is less, your urine will become more concentrated and the volume also will be less. And that's because ADH and aldosterone will be produced. So this kind of is really a review and tying in with whatever you did much earlier. The electrolyte balance, which is sodium and potassium, calcium, all of this is regulated by hormones, remember, which act at the distal convoluted tubule, the collecting ducts, and the distal part of the ascending limb of the loop of Henle. And then we'll see the pH of blood, which we'll be doing, how the pH also maintains electrolyte balance, because depending on whether you have acidosis or you have alkalosis. So here this is just showing you what we just went over, regulation of water intake and output. So imagine that you have decreased plasma volume, so your blood pressure will decrease. The kidney, the cells in the kidney, which are in the afferent arteriole, secrete renin, which goes through the renin-angiotensin mechanism, will produce angiotensin II, and that will act by itself. Angiotensin II will act on the hypothalamic center. There'll be a sensation of thirst. You want to drink that. The water moistens your throat and your stomach. It's absorbed. You drink it. It's absorbed from the GI tract, and that increases your plasma volume and your osmolarity comes down. And when you drink water, that kind of quenches your thirst, so you don't kind of keep on drinking that too much. On the other hand, look at this. If there's increased osmolarity and sodium concentration is more, this goes through and stimulates the osmoreceptors, and this causes ADH to be stimulated. So this is pretty much what we did earlier. So on this side, it shows you how ADH is secreted. On this side, through the renin-angiotensin mechanism, how aldosterone will be secreted, and that will kind of regulate how much urine will be passed out depending on whether you drink more or you have less. Okay, so answer this, and I mentioned this earlier. Why does this happen? This is extracellular compartment, and this is intracellular compartment. ICC and ECC stand for that. Okay, the correct answer is the fluid moves from the intracellular compartment to the extracellular compartment. Okay, here, that's why they look dehydrated. Remember, the person is dehydrated. So let's go back here. Look at this. When the person is dehydrated, they have lost fluid, right? The fluid, you're dehydrated because you've been vomiting, you've either lost blood, or you have diarrhea, you're sweating excessively, all that is getting out from the extracellular compartment, right? So what happens then? The fluid has gone out more. So in the extracellular compartment, the solutes are more because more fluid has gone out compared to solutes. So extracellular compartment becomes hypertonic. Where is it going to get water from? It's going to pull the water from the cells. When the cells push water out, they shrink. Because they shrink, that's why your face and everything looks, near your eyes, that area looks shrunken, depressed, your skin turgor or elasticity is gone. That's because the cells have shrunk. If the cells were well hydrated, your face and everything else would look normal. Okay? So you must understand this very, very clearly. And just to show you this, so this, if you go back, always go back to this picture. This is a really nice picture. When you're vomiting, you are dehydrated, you are having blood loss, hemorrhage, all of that is first going out from here. When it goes out from here, automatically fluid has gone out, so the osmotic pressure in this rises. Osmotic pressure in this rises, it's going to pull fluid from here into the cell. So this now, when it pushes fluid, it loses fluid here, now it becomes hypertonic because the solutes here are more compared to the water. This is hypertonic, it's going to pull fluid from the cells. So the cells will shrink. Okay, they will look much smaller. Have you followed that? If you drink fluid, that will go first into the bloodstream. From the bloodstream, now what happens? The solutes become diluted, so osmotic pressure is much less. So more fluid kind of remains on the outside. Now this here gets diluted, so there's more fluid here which pushes it into the cell. So now the cells will swell. So that's when you look edematous, if you drink too much fluid or you have too much IV fluids pushed inside you. Okay? Okay, let's look at acid-base balance. The pH of blood is dependent on the free hydrogen ion concentration in the blood. It has to be free. It should not be attached to anything. We like to keep the pH of blood between 7.35 to 7.45. So it has a range, and the blood pH, you like to keep it between that. And the body does everything to maintain it in that range. Normally, we take 7 as the neutral position. Anything which is below 7, from 0 to 7, we say that is acidic. That means acidic is a low pH because the number is low. But this means that the amount of hydrogen ions, free hydrogen ions, is more. Do you understand? A low number means the number of hydrogen ions is more. So there is this opposite correlation between them. Okay? In other words, if the pH is 2, that would mean that the number of hydrogen ions is much more. Can you see that? If the pH number is low, that means the number of hydrogen ions is more. If your pH number is high, which is between 7 to 14, it's alkaline or basic. So when we say high pH, I mean the high number. So if your pH is, for example, 9, this would mean that the amount of hydrogen ions is less. That's why your number has become high, your alkaline. Have you followed that? This opposite relationship between the two. So in our body, we have various buffer systems, and they resist these changes in pH. So if your pH is towards the acid, what they try to do is they kind of try to remove the hydrogen ions and bind to it. So if they remove hydrogen ions and bind to it, the amount of free hydrogen ions will be less, so your pH automatically will be adjusted. On the other hand, if your pH becomes high, that means it becomes alkaline. What buffers will do is, if they have hydrogen ions attached to them, they'll give up those hydrogen ions. So the amount of free hydrogen ions will increase, so again your pH will be adjusted. Okay, have you followed that? So we have chemical acid-base buffers. There's one called the bicarbonate buffer system, and we saw this when we were doing the respiratory system. In the kidneys, we have something called sodium bicarbonate as well. Then hemoglobin is also a buffer. Hemoglobin is a protein. So hemoglobin has the capacity to attach to hydrogen ions or release hydrogen ions. So you can see based on what the property is, what the pH is, it will either attach to more hydrogen ions or it will give up hydrogen ions to kind of adjust the pH. And then in the kidney, we also have this phosphate buffer system. So we'll take a look at this. So we have both the respiratory systems and the kidney, the renal system, adjust the pH balance. Now, if the respiratory system is damaged, then obviously the respiratory system cannot work in adjusting pH, okay? So we have two conditions. So for now, don't kind of look at this. So understand this. So we have conditions which are called acidosis and conditions called alkalosis. This could be of respiratory origin or it could be of metabolic origin. Both of them could either be of respiratory origin or metabolic origin. So look at this situation when you have respiratory acidosis. Let's say, for example, somebody is not breathing out normally. They are hypoventilating. Say they have a problem. Their respiratory center is damaged. They're not breathing out normally. Okay, so their normal respiratory rate is low. The respiratory rate is between 12 to 14 or a little bit more, say 15. So if their respiratory rate is less, let's say they are hypoventilating, what will happen? Carbon dioxide will remain in the blood, and carbon dioxide gives rise to hydrogen ions. So can you see that they will end up with something called respiratory acidosis? On the other hand, if somebody hyperventilates, so they breathe out more, their respiratory rate is more than normal, okay? So they are breathing out very, very rapid. They go into a panic attack, suppose, and they keep breathing out very, very rapidly for very long. So what happens? Because they are hyperventilating, they're pushing out a lot of the carbon dioxide. Carbon dioxide finally gives rise to hydrogen ions through this reaction, which you should remember. Water is always present between the cells. It reacts with carbon dioxide, gives rise to carbonic acid, which breaks up into which breaks up into hydrogen ions and bicarbonate ions. Okay? So the more the CO2 in the respiratory system, if you hypoventilate, what will happen? The more hydrogen ions will remain inside, right? If you keep pushing out this carbon dioxide, you are indirectly removing hydrogen ions. So the amount of hydrogen ions remaining in the body will be less. So you'll end up with alkalosis. So you'll get what is called respiratory alkalosis. So anything which is of respiratory origin, obviously the respiratory system is responsible for creating the problem, acidosis or alkalosis. So the kidneys will take charge of it. So if there is acidosis, what does the kidney want to do? It wants to get rid of hydrogen ions. So we'll find that hydrogen ions somehow will pass out in the urine. If, on the other hand, you have a situation where you have respiratory alkalosis, what would the kidneys want to do in this case? Would they want to throw out hydrogen ions in the urine, or would they rather throw out bicarbonate ions and actually preserve the hydrogen ions? Preserve the hydrogen ions. So they'll want to throw out bicarbonate. Okay, so that's how the kidneys will react. Let's take a situation where the respiratory system is normal and nothing's wrong with the respiratory system, but you have acidosis or alkalosis, and that is of metabolic origin. So imagine someone who is exercising a lot, like running for a really, really long time, okay, or doing some sort of exercise of 4 hours. So since your tissues are metabolizing, what happens when tissues metabolize? They produce more carbon dioxide, right? When you produce more carbon dioxide, you will produce hydrogen ions. So you could go into a state of acidosis. You understand? So the respiratory system will try to take care of it, and the kidneys also will try to take care of it. How will the respiratory system try to take care of it if you are exercising a lot and you're going to be producing too much carbon dioxide and you end up with more hydrogen ions? What do you think would be the respiratory system way of compensating for this? Think what happens when you exercise. Does your breathing rate? Yes, exactly. Hyperventilate, right? If you hyperventilate, what will happen? Whatever extra carbon dioxide your tissues are producing, you're blowing that all off. So you don't allow the hydrogen ions to increase in the blood, the free hydrogen ions. And that you might have seen when you exercise, when you run, your respiratory rate starts going up, right? Because not only do you want to take in more oxygen to provide your tissues with that, you also want to get rid of the excess carbon dioxide. And the kidneys, of course, they take some time to do their job. They're not as fast as the respiratory system. So in any case of acidosis, the kidneys will get rid of hydrogen ions in the urine, okay? Let's look at a case of metabolic acidosis. Oh, I just did, sorry, metabolic acidosis. Let's look at a case of metabolic alkalosis. Alkalosis will occur when the amount of hydrogen ions is less. So metabolic alkalosis could occur if someone is vomiting. When you vomit, what happens? The fluid and food or whatever comes out from your stomach. Your stomach produces acid, hydrochloric acid. So when you vomit, hydrochloric acid comes out in the vomitus. So you're losing hydrogen ions. Can you see the... So when you lose hydrogen ions, they are less in the body and end up with a situation of alkalosis. How would the how would the respiratory system compensate for this? Hypo, you want to breathe less so that you preserve that. And you might have noticed next time hopefully you won't, but whenever you vomit, try and look at it and see whether your rate, in the beginning, yes, the exertion might cause the excessive hyperventilation, but later you will see that, the respiratory rate kind of comes down a little bit, okay? And the kidneys will compensate if the thing continues by making sure that they preserve the hydrogen and remove the bicarbonate. Yeah, you had a question? So here we can see that if you look here, so the respiratory system will compensate in metabolic acidosis and alkalosis. The renal system will compensate in the respiratory acidosis and alkalosis as well as metabolic. But you can see that whenever alkalosis or acidosis is of respiratory origin, the respiratory system can't compensate because it's the one which is actually causing the problem, right? So let's take a look at what happens. So let's look at how hemoglobin acts as a buffer system. And this is what you did when we were doing the respiratory system. So kind of a bit of a review. So let's say here are the cells in the tissues which are metabolizing, producing carbon dioxide. This carbon dioxide, remember, across the respiratory membrane, the carbon dioxide diffuses into the blood, and oxygen from the blood comes in through the respiratory membrane to go into the tissues. That oxygen which came in the blood, remember that oxygen was bound to hemoglobin. 98% of oxygen is bound to hemoglobin. Only 2% is dissolved in plasma. You remember that from respiratory? If you don't, I'm reminding you now. Okay. So this oxyhemoglobin, when it comes towards the tissues, the hemoglobin gives up the oxygen and remains as free hemoglobin. That oxygen diffuses across and goes into the tissues. The tissues now have produced a lot of carbon dioxide. Some of the carbon dioxide goes into the plasma and gets dissolved as carbon dioxide. Some of the carbon dioxide goes into the plasma where through a slow reaction it binds to water in the plasma, forms carbonic acid, and which breaks up into bicarbonate and hydrogen ions. These hydrogen ions, if they were left free, what would happen? Acidosis would occur, right? So these hydrogen ions are bound to plasma proteins. So that's how the body normally prevents the free hydrogen ion level from rising. Most of the carbon dioxide gets into the red blood cell where you have this enzyme called carbonic anhydrase. So same reaction but faster because of this enzyme, forms carbonic acid. Bicarbonate and hydrogen ions are formed. Look at this hemoglobin which was liberated here. It now mops up these hydrogen ions and forms reduced hydrogen hemoglobin. So hydrogen ion levels are not allowed to rise. And that's how bicarbonate goes out. And if you remember, 70% of carbon dioxide travels as bicarbonate ions in the plasma. Imagine if the tissues were metabolizing a lot. They were producing a lot of carbon dioxide. As these hydrogen ions would be formed, there's only so much plasma proteins that could bind to them. There's only so much hemoglobin that could bind to the excessive hydrogen ions. So The remaining, which do not find any plasma proteins or hemoglobins to bind to them, they will remain free, which is why you end up with acidosis. Okay? Now here, look at what happens. This same blood now, which has got carbon dioxide and all, is coming towards the alveoli. The carbon dioxide, which is dissolved in plasma, goes right across the respiratory membrane. This plasma proteins release the hydrogen ions, which binds to the bicarbonate, that forms carbon dioxide and goes out this way. And the hemoglobin, which was attached to the carbon dioxide, that kind of breaks up, and then you get the same reaction going out. So the more the tissues are metabolizing, the more that is being carried like this. So if you keep blowing out excessive carbon dioxide by hyperventilating, you're preventing the rise of these hydrogen ions. And that's how the respiratory system will take care of it. Okay? Let's look at what happens in the kidney. So if you read through this, I'm not going to kind of read through that because this is just there for you to read. So the same reaction, if you look, happens in the kidney as well. So look at in the kidney, from the blood, carbon dioxide enters into the kidney cell. This forms carbonic acid, which breaks up into bicarbonate and hydrogen ions. If there is more of this present here in the blood, more hydrogen ions are formed. These hydrogen ions are secreted into the kidney tubule, where they meet bicarbonate, which is present in the filtrate. And you can see here then that same reaction occurs in the lumen as well. So it forms water and carbon dioxide. The carbon dioxide gets back into the cell and water goes out in the urine. So you can see that hydrogen ions are removed in this way. So they are secreted into the lumen and then they are removed this way by the kidney. So the more the hydrogen ions, as in the case of acidosis, this is how the kidneys will take care of it. Now this is what happens in the proximal convoluted tubule, where you have enough bicarbonate present. By the time you come to the distal convoluted tubule, most of the bicarbonate has kind of been reabsorbed. So there is less. So now we have another buffer system, which is hydrogen phosphate. So here, if the kidneys secrete hydrogen into the lumen of the tubule, it will form dihydrogen phosphate, and that's how what will pass out in the urine. So you can see how bicarbonate and hydrogen phosphate act as buffers, and they attach to this hydrogen ion and pass it out in the urine. So the more, if there's a case of acidosis, you can see hydrogen ions will be secreted. If there was a case of alkalosis, then what will happen? It won't secrete hydrogen ions. Instead, it will secrete bicarbonate. It will do the opposite, just to make sure that the pH is adjusted. Okay? So let's take a look at this question. It is both, yes, it is both. And remember this, and this is a concept you must remember. When I say pH falls, I'm talking about the pH number, the way you would express it, between 0 to 14. So in acidosis, it is anything between 0 to 7. So when someone's pH is 7.35, if it falls, then the number falls, okay? So the pH can become 7.2, or it can become 6.2 or 6 or whatever. But this falling is because of hydrogen ion concentration increasing in the blood. So increased hydrogen ion concentration leads to a decreased number as far as pH is concerned. So please remember this thing. So this is what we saw in that slide up there, how the respiratory system regulates acid-base balance. So this reaction we saw, carbon dioxide reacts with water. The reaction can go either way depending on which way you want it to go. Forms carbonic acid, which breaks up into hydrogen ions and bicarbonate ions. So if there's excessive metabolism, you're going to form more carbon dioxide. This will lead to more hydrogen ions, so automatically you'll get acidosis. To compensate for this, if the respiratory system hyperventilated, what happens? You're pushing the reaction this way. If you keep removing carbon dioxide as fast as hydrogen ions are formed, you're going to be able to adjust the pH, which is what happens when people exercise and they begin to hyperventilate. If there's reduced metabolism or excessive loss of fluids, reduced metabolism, for example, when you're sleeping, you don't hyperventilate, your respiratory rate comes down. As I said, and excessive loss of fluids, say when you vomit, so you're losing hydrochloric acid. So there's less hydrogen ions, and the respiratory system will be able to save the hydrogen ions if it doesn't push out carbon dioxide. So if it hypoventilates, you kind of keep whatever hydrogen ions are there, you keep that back in the blood, so you raise the amount, and so you bring the pH back to normal. Okay? The renal regulation, as we saw there, this will compensate when the respiratory system is at fault. It will also compensate in metabolic situations as well. So it's not that it only compensates when the respiratory system is at fault, but it will compensate in metabolic systems as well. The renal system takes a bit longer to compensate compared to the respiratory system. So let's say someone is, so this here the cause is because of the respiratory system. So suppose someone is hyperventilating, like they're being stressed, too much pain, whatever the reason may be. So too much of carbon dioxide leaves the body, which means that there is less hydrogen ion, so you enter into a state of alkalosis, right? So the kidneys will compensate by pushing out bicarbonate, so they don't reclaim it, bicarbonate goes out, and they keep the hydrogen ions in. So they reabsorb more hydrogen ions. So that's what the kidneys will do. So this situation leads to what is called respiratory alkalosis. On the other hand, as in sleep or for any other condition, there is a problem, let's say, with the respiratory center. If a person is hypoventilating, carbon dioxide is retained in the body. Through that reaction, hydrogen ions will increase. So this will lead to a state of respiratory acidosis. So it's acidosis. You have more hydrogen ions. You want to get rid of it. So what the kidneys will do is they'll secrete the hydrogen ions into the lumen, where you saw it will either react with bicarbonate as the buffer or hydrogen phosphate. So it will pass out in the urine, and they'll retain bicarbonate. So hydrogen ions will go into the lumen and pass out in the urine, and they will reabsorb more bicarbonate, and again adjusting the pH. Okay? So let's say, again, something I asked you, so let's review these areas. So if a metabolism increases, what's the best way for the respiratory system to maintain homeostasis? Increase respiratory rate. Yes, if your metabolism increases, you're producing more carbon dioxide. More carbon dioxide means you're going to produce more hydrogen ions. You don't always have to be exercising. In case of type 1 diabetes, if you remember from when we did the endocrine system, remember fats were broken down because in type 1 diabetes they cannot produce insulin. So they break fats and proteins in order to produce ATP. When fats are broken down, it produces fatty acids which lead to something known as ketone bodies, which are fatty acids. So this is an acid, right? So if it's an acid, that means acids give off hydrogen ions. So a person with type 1 diabetes goes into diabetic acidosis. So if you observe a person who goes into this acidotic state, these people actually hyperventilate, and it's a special type of breathing that you see, which is called Kussmaul's respiration. So they hyperventilate because of that increased hydrogen ion. So the way to get rid of that increased hydrogen ion is, if you hyperventilate, you get rid of carbon dioxide, which pulls hydrogen ions, through that reaction. Okay, so this is very, very typical of a person of type 1 diabetes. So if a person hypoventilates, what would be the best way for the kidneys to maintain pH balance? So think what happens when a person hypoventilates. What would it give rise to? And then what would the kidneys do? Okay. Now I'm telling you here that the person is hypoventilating. Okay, that means there's a respiratory problem. So the problem is in the respiratory system. If a person hypoventilates, what will rise in the blood? Carbon dioxide will rise in the blood, which means that the end result will be hydrogen ions will increase, right? So they will end up in a state of what we will call respiratory acidosis, right? If there's respiratory acidosis, the amount of hydrogen ions is more. What should the kidney do? Get rid of it. So what should it do? It will secrete hydrogen ions into the filtrate, okay? So when you get these kind of questions, first start thinking of what is written here. What will happen because of that? Like in the previous question, if you look here, the way you thought about this, the previous question, here you said metabolism increased. When the metabolism increases, you're producing more carbon dioxide, right? And if you're producing more carbon dioxide, you could end up with something known as metabolic acidosis. So now the way the respiratory, what you want to do, you want to get rid of hydrogen ions, and the way the respiratory system will do is will hyperventilate. Now here, the next question was, I'm saying that the problem is with the respiratory system. I'm saying the person is hypoventilating. So then I'm asking you what the kidneys will do. So when you read the question, kind of read it in two steps. First, determine what's going to happen because of this, and then what the kidneys or the respiratory system is going to do. Okay?